Hello, my name is Helga Stopper and I'm a professor for toxicology at the Department of Pharmacology and Toxicology at the University of Würzburg in Germany. And this lecture is about the mutagenicity of natural plant products. In this lecture, I will first give a short introduction, and then during the lecture, at 2 points, I will pose questions and give literature recommendations that you can then look at for interactive learning. I would estimate that each of these activities can take about an hour. I will then provide the answers for these questions in the lecture. So-called botanicals are natural plant-derived products. Natural plant-derived preparations, as well as some herbal spices, are heavily advertised as health-supportive products without side effects because they come from nature. Some of the claimed positive effects are antioxidant, anti-inflammatory, and anti-carcinogenic activities. Therefore, about 20% of Americans and a similar percentage of Europeans are regularly supplementing their nutrition with botanical products. But we have to be aware that plants make chemicals for communication and for defense, for example, against herbivores, plant pests, and sunlight. There are many examples of plants which cause severe and even life-threatening acute toxicity, such as belladonna or autumn crocus, and these are well known. And it's also well known that even some food items cannot be consumed uncooked because of their natural toxicity. For example, kidney beans or lima beans. However, mutagenicity and, as a possible result, carcinogenicity is much more difficult to detect because it may not be associated with any acute symptoms. The development of cancer has a latency time of years and up to decades after exposure. But there are some known examples for carcinogenic natural substances in plants. And this is where the first task comes in, and you might plan about 1 hour for reading this literature. So based on published literature, can you find an example for a carcinogenic natural plant constituent and briefly describe the history of its identification? And I've listed a recommendation for a publication where you can find the information. So you can pause here for reading of the literature to answer the question, and the following slides will then provide the answers to the question. So here are the answers for task 1. The publication that I have recommended was Aristolochic Acid-Associated Cancers, a Public Health Risk in Need of Global Action. And it's from the year 2022. It describes evidence for mutagenicity and carcinogenicity of aristolochic acid. And it suggests prevention measures to reduce the aristolochic acid-related cancer incidence. So let's look at the history of aristolochic acid-related findings. In the 1960s, first hypotheses linked aristolochic acid to cancer. The Balkan endemic nephropathy, a renal disease first described in areas around the Danube River, often leads to the development of urothelial carcinoma. It is thought that aristolochic acid-producing wheat contaminated the flour in this region. The next observation was made in the 1990s when Belgian women who had consumed an herbal product for weight reduction experienced kidney failure. The product erroneously contained an aristolochic acid-producing herb. In 39 of these patients, upper tract urothelial neoplasia was found in 18 of them and urothelial dysplasia in 19 of them. Between 2008 and 2010, several investigations linked aristolochic acid-containing herb consumption in Taiwan to an increased risk of urinary tract cancer, kidney failure, and adrenal cell carcinoma. A mutational signal for aristolochic acid-exposed cells or tissue was identified around 2012. It can be detected in aristolochic acid-induced tumors and therefore yields evidence for aristolochic acid as an inducing agent. The case of aristolochic acid shows that there is a need for identification of mutagenic and carcinogenic natural plant constituents. We have in vivo and in vitro mutagenicity testing methods available for the approval of chemicals and pharmaceuticals. Can we also use them for testing substances that are natural plant constituents? Well, there are problems. First of all, across all plants, it was estimated that they produce between 100,000 and 1 million metabolites. And any single plant species probably produces 5,000 to tens of thousands metabolites. These substances are required by plants to provide them with stress resistance, for example, protection against herbivores, or adaptation to environmental conditions such as UV light. So there's no way that many substances can be tested using available methods. And there are more complications for testing individual substances from plants. A good natural constituent may inactivate a bad constituent. Licorice is a hepatoprotective herbal medicine. It is often used together with pyrrolizidine alkaloid containing herbs in traditional Chinese medicine formulas. Many pyrrolizidine alkaloids are toxic and mutagenic, predominantly causing liver damage and cancer in the liver. However, if PA are combined with licorice, no poisoning cases or liver damage in experimental animals has been observed. This seems to be due to an inhibition of metabolic activation of PA by the active compound in licorice. And there are even more difficulties in vitro testing. Metabolism. Hepatocytes activate certain toxic substances in the liver, but the target cells or tissues are sometimes not the hepatocyte or the liver itself. We can introduce metabolic activation in our current in vitro models, but this does not represent human liver metabolism completely. Systems which combine activating hepatocytes and potential target tissues are still under development and are not used at this point in standard routine testing. This is not a specific problem for botanicals, but also applies to them. Then the nonlinear dose response. Some beneficial substances, for example some natural antioxidants, become mutagenic in vitro at higher concentrations. That is, they then exert oxidative effects while they were antioxidative at lower concentrations. It is difficult to determine which consumption limit ensures that this turning point from protective to harmful is not reached in vivo in humans. This is also not specific for botanicals, but occurs often in the context of natural plant components. And then there's the potential problem of glycosides. Many substances are present in plants as glycosides. That means they are bound to sugar molecules. It is not well known to which extent that affects the uptake in the gut or the uptake into target cells. Some compounds may need to be deconjugated, so cut loose from the sugar, by colonic bacteria for uptake. Others may not need this step. If certain glycosides do not enter cells in vitro test systems, but are deconjugated in the colon and taken up by the body as aglycones without the sugar molecule, we may not be able to find the mutagenic ones among them as positives in our test systems. Therefore, testing of natural plant constituents cannot be done as single substance testing. Complete botanical preparations have to be tested. These are complex mixtures, and it has to be clarified whether current testing strategies are effective and sensitive enough. A consortium has formed to guide the development of a suitable testing strategy, and this is where task 2 comes in. And again, I would like you to plan about 1 hour for this. So the task is to read about the work of the Botanical Safety Consortium and briefly describe their current plans for the development of a testing strategy for botanicals. And there's a literature reference for that and also an internet site where you can look at. And again, you can pause here for a reading of the literature and doing some internet research to answer the question, and then the following slides will provide an answer to this question. The goal of the Botanical Safety Consortium is to identify a suitable set of methods for testing of botanicals. So far, botanicals have been selected representing different aspects of toxicity, for example genotoxicity, but also hepatocardio, neurosystemic development and reproductive toxicity. Literature reviews and chemical characterization of the extracts have been performed. In silico analysis of known components will be provided. Working groups select methods for each toxicity endpoint, which are most likely suitable for the purpose of testing botanicals. Then botanical case studies with a few selected botanicals will provide experimental results with the chosen methods. Then the selection of methods will be discussed, modified if needed, and applied to a larger number of selected botanicals. Possible additional methods for elucidation of mechanisms of substance toxicity may be added at this point. In the end, a testing strategy recommendation will be established. So, here's the take-home message. Substances within botanicals and plant preparations and herbal spices can be mutagenic and may cause cancer. Substances within botanicals cannot be tested individually because there are too many. Other substances in the same plant may inactivate the toxic compounds. Substances known as protective may be toxic or genotoxic only at high concentrations. Knowledge about the contribution of glycosides is limited. They may be biologically active and may or may not be detectable in standard testing. New strategies are needed and are being developed currently to elucidate potential health risks associated with the consumption of botanicals. And with this, I would like to thank you for participating in this activity.